The problem that we will discuss in this presentation is with regards to the mysterious unhealthy supplement that caused the death of a man. Research has shown that the disruption of mitochondrial ATP synthesis can cause cell death by either apoptosis or necrosis, since ATP is required for the completion of apoptotic programming. Small decreases in ATP will cause the cells to die via apoptosis, whereas large decreases will cause death via necrosis. From this situation given to us, it is known that the ingestion of a protein powder with an unknown supplement caused an increase in expired carbon dioxide, oxygen, and muscle weakness, indicating a lack of ATP production, which correlates with death. However, the mechanism by which metabolism is inhibited by the addition of this protein supplement is unknown, presenting a knowledge void. We speculate that nitric oxide competes with oxygen for the binding site on cytochrome C oxidase, decreasing oxygen's ability to oxidize the electron transport chain and to produce a proton motive force for ATP production. Our purpose is to identify the supplement that killed the man so that we can provide insight to the dangers of the ingestion of this supplement.
This is the overarching model of ATP synthesis and metabolism. The green arrows in our flow model represent the activation of enzymes by substrates. We included the process of glycolysis and TCA in our model to demonstrate how it interacts with the electron transport chain by providing the reducing agents needed for the electron transport chain, such as NADH and FADH2. There are many inflows and outflows in our model. However, for this problem, we will be primarily focusing on the electron transport chain. The regulated mass in the electron transport chain is ATP. Inflow to the ETC is NADH, FADH2, and oxygen. The oxidation of NADH and FADH2 reduces the electron transport chain and allows the transport of hydrogen ions from the mitochondrial matrix to the intermembrane space, creating a proton motive force. Oxygen is reduced at subunit 4 of the ETC, cytochrome C oxidase, oxidizing the ETC. Following oxidation, ATP synthase uses the proton motive force to drive the synthesis of ATP. Outflow of ATP happens during exercise when we are using ATP for energy, increasing the pools for ADP and AMP. For our problem statement, this is our mass balance model. The regulated mass in our problem is ATP. The inflow for this mass is NADH, FADH2, O2, ADP, and PI. These substrates are provided by glycolysis, TCA, and respiration. The conductance is through the redox reactions through the electron transport chain. The outflow for ATP is ADP and AMP, and the conductance for this is ATPases. The sensor and the controller in this situation is the pool of ADP and AMP because they activate GFOS and PFK in glycolysis, increasing the conductance for the inflow of NADH and FADH2.
From the problem given to us, it can be seen that after the man ingested the protein supplement, oxygen and carbon dioxide increased in his expired air. Additionally, before collapsing, he complained of muscle weakness, which is caused by a decrease in ATP levels.
The study that was used to test our hypothesis is a paper from 1994 by Byrne. This study investigated the effects of nitric oxide on cellular respiration through light-activated release of nitric oxide from nitroprusside. The study isolated samples of cytochrome C oxidase from the nervous tissue of rats. The samples were then exposed to nitroprusside. When presented with light stimulus, nitroprusside releases nitric oxide. In this study, oxygen consumption was measured using an oxygen-sensitive electrode. They found that as light exposure increased, the concentration of nitric oxide increased and cellular respiration decreased, as demonstrated by a decreased consumption of oxygen. They also found that removing the light post-exposure resulted in a decreased pool of nitric oxide and respiration rate increased. Data shows that there is a decreased rate of respiration associated with an increased concentration of nitric oxide. The study suggests that nitric oxide more readily binds to cytochrome C relative to oxygen. This supports our hypothesis that nitric oxide competes with oxygen, resulting in less binding sites for oxygen to oxidize the electron transport chain. This reduces conductance of the ETC, resulting in an inability to further accept electrons and produce a sufficient proton motive force. This decreases the inflow to the hydrogen pool in the intermembranous space and eventually decreases the rate of inflow to ATP that will eventually lead to death.
Based on this study, we have added nitric oxide as controller in this mass balance model. Nitric oxide has a role in the synthesis of ATP as it inhibits the oxidation of cytochrome C oxidase and the electron transport chain, decreasing the conductance for inflow of ATP. As a result, the pool for NADH, FADH2, oxygen, ADP, and PI increase. This introduces another regulating variable for ATP inflow in addition to our original hypothesis.
Nitric oxide more readily binds to cytochrome C, and oxygen is less able to bind and act as the final electron acceptor. Therefore, the ETC cannot be oxidized. This results in an increase in oxygen concentration and decrease in flow to ATP. This leads to increased inflow to ADP and AMP pools, which activate conductance through glycolysis via GFOS and PFK. Increased flow through glycolysis increases P1 for the tricarboxylic acid cycle, acetyl-CoA. Increasing P1 increases flow through the TCA, increasing the production of carbon dioxide.